In this video presentation, we will discuss about the Bluetongue, one of the important notifiable disease, and a vector-borne viral disease, affecting sheep. These are the notifiable diseases affecting sheep and goat, listed by OIE. Bluetongue is also one of the notifiable diseases listed under Classification. Reoviridae family, classified under the Baltimore Group 3. Group 3 comprises of viruses with double-stranded RNA genome. The word Reo refers to respiratory enteric-orphan viruses, that is, these viruses infect humans, respiratory and intestinal tracts without symptoms. Some of the veterinary important viral diseases under this family are Bluetongue and African horse sickness, under the genus Orbivirus. Rotaviral enteritis, under the genus Rotavirus. The diseases under the genus Orbivirus and Cultivirus are Arthropod Borne viral diseases. The diseases under the genus Rotavirus and Reoviridae are non-arthropod borne viral diseases, transmitted through fecal-oral route transmission. In this lecture we will discuss in detail on Bluetongue. The disease name refers to its clinical feature, cyanosis of tongue. The other names for this infection are sore muzzle and muzzle disease. The causative agent of disease is Bluetongue virus, which belongs to genus Orbivirus, of the family Reoviridae. This virus is an arbovirus, that is, transmitted by biting midges arthropod, like CULTIVATION. So the infection is a non-contagious, arthropod-borne viral disease, characterized by fever, oral ulcers, swelling of lips, tongue, inflammation of coronary band, and lameness affecting sheep. Host range. Apart from sheep, this virus also infects goat, antelope, elk, camel, cattle, and buffalo. This infection is highly susceptible for sheep's. Mortality rates can vary, 30 to 80%. In cattle, it causes an inapparent infection. This infection is prevalent in areas wherever the CULTIVATION vector is present, like Africa, America, Australia, some countries of Southern Asia, and Oceania. Currently, there are 27 Bluetongue virus serotypes existing worldwide, classified based on the variations in their VP2 outer capsid protein. So these VP2 proteins are serotype-specific antigen. Similarly, there are 14 Bluetongue virus serogroups existing worldwide, classified based on the variations in their VP7 core capsid protein. So these VP7 proteins are serogroup-specific antigen. In India, the first Bluetongue outbreak was reported in 1964, at Maharashtra. In India, till date, 13 Bluetongue serotypes have been isolated, mostly from southern Indian states. But vaccination is done using inactivated pentavalent vaccine against 5 serotypes, serotype 1, 2, 10, 16, and serotype 23. Virus morphology. This virus possesses triple spherical capsid, layered one over the other. Outer, core, and inner capsid, which encases segmented genome. This segmentation may leads to genetic reassortment. The entire virus is about 75 to 80 nanometer diameter. VP2 and VP5 codes for the outer capsid. VP2 is a serotype-specific antigen, and they play a major role in the antigenicity, that is virus-neutralizing antigens. VP3 and VP7 codes for the core capsid. VP7 is a serogroup-specific antigen. And VP1, VP4 and VP6 codes for the inner capsid. Genomic Organization. The genome is multipartite, that is segmented, double-stranded RNA. This segmented genome may result in genetic reassortment. The Orbivirus possess 10 segments. Rotavirus possess 11 segments. Cultivirus, 12 segments. And Reoviridae, 10 segments. So, our interest here is on Orbivirus, which possess 10 segments. Each segment codes for 1 or 2 proteins. The 5' end is methylated, that is capped. And the 3' end has no poly-A tail. As we already discussed, the segmented genome may result in genetic reassortment. That may lead to evolution of new virus. If a sheep is infected with 3 similar type of viruses, but different serotypes at same time, due to reassortment of segments, that is genetic recombination, there may be evolution of new virus, that may be a hybrid of this 3 virus. This type of major change in the virus is called as antigenic shift. This happens due to reassortment of segments, that is genetic recombination, leads to evolution of new virus. This virus can hemagglutinate mammalian RBCs. In Bluetongue virus, this VP2 outer capsid protein R generally exists as an inactive precursor. But once this inactive protein is reacted to the proteolytic enzymes, The inactive VP2 outer capsid protein precursor, get cleaved or fragmented into active subunits. In this process, the infectivity of the virus gets increased for CULTIVATION insect. Virus replication. The Bluetongue virus enters the host by endocytosis. Following entry, the virus replicates in the cytoplasm. Here, the genome is double-stranded RNA. Only, the negative sense RNA are transcribed and synthesis the positive sense RNA. This positive sense RNA, which are similar to mRNA, are translated for viral proteins. These positive sense RNA are synthesized in excess. So, on the other side, these excess positive sense RNA act as template for negative sense strand synthesis. Later, these synthesized double-stranded viral RNA and the translated viral proteins self-assembles to form virion. Physical and chemical inactivation. This virus get inactivated at the temperature of 50 degrees Celsius for 3 hours, or at the temperature of 60 degrees Celsius for 15 minutes. These viruses are also sensitive at the pH less than 6, or at the pH more than 8. This virus is inactivated by the chemicals like beta-propiolactone, iodophores and phenolic compounds. Transmission. This is an arthropod borne viral infection. So biting midges like CULTIVATION SPP, blood and semen from the infected animal, can act as the principal source of this virus. This virus enters the host through infected midge bite, which is a biological vector. The incubation is on an average of 5 to 10 days from the entry of virus. Pathogenesis. Following entry of virus through infected midge bite, the virus does its initial replication in the regional lymph node, lymphoreticular tissues, blood vessels. Followed by viremia, the virus get distributed to entire body, and panleukopenia. This infection is noticed as acute, or inapparent form. Inapparent infection is seen in cattle and other animal species, for certain serotypes. Acute infection is seen in sheep, which is characterized by high fever, excessive salivation, swelling of face, tongue, cyanosis of tongue, inflammation of coronary band, that is region above the hoof, that may lead to lameness, and abortion are noticed in pregnant ewes. The picture showing the swollen face and tongue, which are cyanotic, in sheep. The picture depicting the inflammation of coronary band, that is, region above the hoof, that may leads to lameness in the animal. Diagnosis can be done in field level or in the laboratories. Field diagnosis. Based on the prevalence of the biological vectors like CULTIVATION species. By signs and clinical symptoms observed like swollen face and tongue, lameness. And by post-mortem findings like ulcerations in digestive and respiratory mucosa. Next. Laboratory diagnosis. For a lab diagnosis, blood, paired serum samples are collected from live animal. In dead animal, spleen, liver, bone marrow, heart blood, lymph nodes, and aborted fetus are collected. These are some of the laboratories in India, performing the small ruminants disease diagnosis. Like, ICAR, Nividi, Bangalore. Kadrad, IVRI, Bareilly. Virology Laboratory, IVRI, Mukteshwar. This virus can be cultivated or isolated in lab by 3 ways. Number 1. Embryonated egg inoculation. 13 to 15 days old embryonated chicken eggs are used. For this, intravenous route of inoculation method is done. The second method of cultivation of this virus is cell culture system. BHK21, varicells, and insect cells can be used. The third method of cultivation of this virus is animal inoculation. Here, sheeps are used. These are some of the laboratory tests can be done for diagnosis of this virus. For antigen detection, immunofluorescence assay, antigen capture ELISA, virus neutralization test, reverse transcriptase PCR, and DNA sequencing is done. For serogrouping the virus, the first 3 mentioned tests is done. For serotyping the virus, virus neutralization test, and DNA sequencing, a VP2 gene is done. For antibody detection, complement fixation test, agar-gel immunodiffusion test, competitive ELISA, and indirect ELISA is done. In these tests, known Bluetongue viral antigen is used. Prevention and control. These are the measures followed for the prevention and control of this infection. Like, vector control, primarily done, here water management is done to reduce of CULTIVATION breeding sites. Use of insecticides and larvicides by spraying over breeding areas. And dipping of animals in insect repellents. Quarantine and restricted susceptible animal movement, during insect activity period. Vaccination using inactivated vaccine, against specific BTV serotypes of concern in a particular area. Serological surveillance, identification and tracing of susceptible and potentially infected animals. For vaccination, an activated pentavalent vaccine is used in India. This vaccine is inactivated by binary ethylenimine inactivating agent. This inactivated vaccine is emulsified with montanide, which is an adjuvant. This vaccine is prepared against 5 serotypes: serotype 1, 2, 10, 16, and serotype 23. These are specific BTV serotypes of concern in India. No specific antiviral drugs are available for this infection. Only symptomatic treatment is done using NSAIDs and antibiotics to prevent bacterial infections is done. With this we are coming to the end of Bluetongue infection. In next video presentation we will discuss on the African horse sickness, under the same genus Orbivirus in detail. Hope the lecture is informative and useful. Thank you.